In Section 6 of the presentation and Chapter 6 of the book, we look at some of the more recent advances in the field of clinical immunology, specifically the advent of monoclonal antibodies, a biologic therapy that's able to specifically, with rifle shot accuracy, destroy specific targets in the cancer and also in the different proteins that allow the cancer to grow, multiply, and spread. So if we look at the timeline a little bit, here's Burnett and Thomas in 57, okay? Virchow, who said that, cancer is a wound that does not heal. Coley back in the 1890s with his combination bacterial toxins with the serratia, etc., that was getting good results. Now, I mentioned earlier that Coley's work was an improvement over work that was done 50 years earlier. 50 years before that, there were medical, and I spent some time in London doing some studying there at one of the university hospitals, and they had books medical textbooks going back to the early 1800s. And one of the doctors who was rather ancient told me about a doctor who was ancient in his time, about a technique that they used to use in the early 1800s of somebody who had an advanced cancer and basically was a hopeless case. And I said, okay, what was this treatment? He said, well, sometimes the treatment worked and sometimes it killed the patient. It usually killed the patient, but if it didn't kill the patient, they went into a spectacular remission. And I said, well, this is interesting. What was the treatment? He said, well, what they did was they went to the patient back to their house. They went out to the barnyard. They took a piece of wood. They stuck it in some cow dung, and they jammed it into their thigh. Oh my God. I thought he was kidding me, but he said no, and he showed me the textbooks, and I actually saw they had pictures, and it was, oh my God. So, if the patient survived the sepsis, then sure enough, there were documented cases of people going into remission from having contaminated wood splinters jammed into their thigh with cow dung. So I would imagine that Coley, being a smart guy and having access to these same medical journals, may have actually come across something like that and tried to figure out what the common denominators were and try to see if he could clean it up a little bit, which I think he cleaned it up quite a lot, much to his credit, and had some very good results and helped far more patients than he killed. So that was a huge step forward. But nonetheless, there were many documented cases of patients whose lives were saved by this attack of the infected wooden splinter. Then they had pictures of the scarring and that occurred. And you can imagine the debility of that.
What's the procedure for that?
Well, that was, okay, so we're up to ICD-9 10 now, okay. I think that was back in the ICD-1 days or something, I don't have any of those references, but it was, nonetheless, it's amazing what happened in the past, but it's also interesting if you look at some of that stuff and look at what came after it, and you can see where the connections are, you can see how things sort of evolve. It all started off by a splinter of wood in the leg, but whatever. So getting back to that, then they took it a step further and they said, okay, if these things are generating an immune response, what is the immune response doing? So they started to look at the different antibodies that were produced, and they started to look at receptor sites. And one thing led to another, and before you knew it, they were able to create monoclonal antibodies that had very specific targets. So this is some of the monoclonal antibodies that are available. These are the names, all right, and this is the format. We'll get to that in a minute. These are the targets. So CD20, okay, is going to knock out some of your more mature B cells, for example. HER2 new, okay, breast cancer, right? Then we have CD33 knocking out leukemia, myeloid leukemia, so on and so forth. Here's VEGF, okay, Avastin, so on and so forth. So they came up, but looking at the literature, there's a clear evolution of thinking of, if something worked, why did it work? And that's probably why in a lot of these articles they go through this whole rehash of what came before them and sort of what led them to think about this next step. So the immunology literature is just fascinating to read in that way. And now we have some very sophisticated monoclonal antibodies that are used for a variety of very specific reasons that we're going to get to. Okay, so a little bit about the monoclonal antibodies. All right, so basically what we have 4 types of monoclonal antibodies. The first one is a 100% murine antibody. Now, murine in medicine means mouse, okay? So when you see murine in the literature, they're talking about mouse. Okay, so the first antibodies were mouse antibodies where they took a mouse, they stuck in an antigen to whatever cancer, had the mouse make a bunch of antibodies against it, gathered up those antibodies, injected it into a person. And why do they do that? They did that because the antibodies would coat the targeting antigens, opsonize it, and create an antibody-dependent cell cytotoxicity target for what was left of the person's immune system to attack. Now, it goes a step beyond that. The step beyond that is that because this antibody was attaching itself to an abnormal protein as they identified it, and because the antibody itself, this is important, because the antibody itself was a foreign protein. It was a mouse antibody, okay? The immune system didn't like it, so it made antibodies against that. So it created this whole big circulating immune complex mess of inflammation and attack against what was in the middle of this whatever this target was. And it sort of worked. Had a lot of side effects. Didn't work so well. So they said, okay, how do we make this better? Well, the answer is you got to get rid of some of the mouse. So then they went from a murine antibody to a chimeric antibody, where they were able to substitute human sequences for some of the mouse sequences here, which made it much less antigenic. Still had the same targeting from the mouse, but it generated much less of a secondary immune response against the treatment itself. And then they got better than that, and they were able to humanize the business end of things. Okay, so we have a humanized antibody, and now ultimately we have 100% human antibodies that we're able to use in treatment. So now the way that they identify these antibodies is by the suffix. So if it's a murine antibody, it ends in OMAB, okay? Like Zavalin, okay? It ends in OMAB. Does everybody see that? Okay. If it's a chimeric antibody, which is this thing, that ends in Zimab, Ximab, okay? So Erbitux, okay? Erbitux, common antibody, right, against epithelial growth factor receptor, right? And that is a chimeric antibody. It's one of these. Humanized antibody, okay? Perjeta, okay? And so it's a Zumab. Everybody see that HER2 target? And now we have ipilimumab, which is a totally human antibody, UMAB. Everybody see that? So when you see the literature and they talk about these different monoclonal antibodies, then you'll know what they're talking about, okay? Now the reason that this is important, okay, this isn't just an academic, I'm trying to make everything clinically relevant here, is the reason that this is important is because the more humanized the antibody is, the less adversely antigenic it will be for a response against the antibody rather than the target is trying to hit. Okay? Does that make sense? Okay? So humanized antibodies are going to be more effective with a better safety profile and better tolerability in general. Okay, this is not a 100% deal. Nothing in medicine is, and certainly nothing in immunology is. But I'll say in general, because especially with ipilimumab, there's some major problems with it, if you're not careful, and that's a totally humanized antibody. But in general, this is kind of the way it works. This represents what percentage of it, more or less, is mouse versus human. The more humanized it is, the better tolerated it is in general. We all good with that before I proceed on? Good? Okay, all right. So we have first generation antibodies, which were totally murine antibodies, which we talked about. And then after that, After that, they have different classes of antibodies. You have first generation, second generation, then we have first class, second class, third class, and this is just telling us how humanized the thing is, and I put the little code across the bottom here because I can never remember it myself. All right. Okay, so what can these antibodies do? Gee, I'm so glad you asked. Okay, how long do we have? Okay, so these antibodies can do a lot of really, really cool things. So these antibodies can, number one, they can block growth factor receptors on a tumor cell. Okay, so that's what Erbitux is doing, for example. It can inhibit angioneogenesis, like Avastin is blocking the ability of cancer cells to grow new blood vessels to feed itself. It can hit other kinds of growth factors, everything from, like, the Herceptin, the whole Herceptin, which are the human epithelial growth factors receptors, and it can also work with synergistically with other monoclonal antibodies, and we'll talk about that in a few minutes. It can block the ability of the growth factors to stimulate the cells. That's one of the ways that Herceptin works. It can activate the complement system, okay? And by activating the complement system, what do you wind up? You wind up with a membrane attack complex. So remember I told you that stuff I was boring you with this morning was going to come back to haunt us. We were talking about complement, we were talking about plasma inflammatory factors, right? Winding up with a membrane attack complex. Well, guess what? One of the things that we talk about and use in our practices takes advantage of that membrane attack complex to kill cancer cells. Okay, other ones like Rituxan can stimulate and trigger apoptosis. And other ones are immune disinhibiting. So there's a lot of different mechanisms. Still, other antibodies can act as a Trojan horse and can carry either a drug, a toxin, or a radioisotope, and specifically target a cancer cell by its ability to specifically connect with it because of the protein structure of the antibody. So these are some of the ways that monoclonal antibodies can be used to attack and destroy cancer cells. And this is a direct outgrowth of that whole lineage I showed you historically, where people made very good observations that something was working with the body to defend itself. Okay, there was the idea of immunosurveillance, and they found that if they did certain things to poke at the immune system, they can get sometimes a positive response. They researched that, they fine-tuned that, and they said, okay, there's certain antibodies that are having this response. All right, how can we manufacture those antibodies? How can we replicate that? One thing leads to the other, and now we have a multi-billion-dollar industry making monoclonal antibodies.
Is that how they take care of Carter's?
Yes. The short answer is these things. That was with a drug called Keytruda, which is a PD-1 inhibitor. We'll get to that. All right. Okay, so what do? Is they take a mouse, okay? They immunize it with what? With specific tumor antigens. Okay, so how do they do that? Well, they take a tumor, how much detail do you want? All right, real quick. So if they're going to make something against a lung cancer, for example, then what they'll do is they'll take normal lung tissue, they'll take diseased lung tissue, they'll macerate it up, they'll break it down to molecular levels, they'll put it onto what's called a 3D dimensional gel electrophoresis. They'll separate out the proteins, they'll see which proteins you have in the cancer cell that you don't have in the normal cells. They'll take those abnormal cancer cell proteins and use those as the antigens that they stimulate the mouse with. Okay, that's it in a real nutshell. What will happen then is that the mouse's immune system will make a whole immune response against this, both humoral and cell-mediated response. They'll then harvest spleen cells from the mouse, okay, that because the spleen will enlarge in the mouse when it's doing a generalized immune response. And what they will do is they will mix them with mouse myeloma cells, okay, which they have in perpetual cultures. And And they do this, there's a number of ways of doing this. The process whereby they're able to mix these two cells is a process called transfection. And there's a dozen different ways of doing transfection. Some of them use viruses, the vast majority do not. None of the techniques that use viruses are currently approved for human use. I guess that movie, I Am Legend, with Bill Smith still resonates in the FDA, so they don't allow any transfection with viruses at this point. So in any case, they transfect the cells, and they wind up with something called a hybridoma. Okay, so hybrid cell, a hybridoma. And the hybridoma is a immortal cell that's producing specific antibodies against whatever antigen the mouse was immunized with. Okay, and they look like generic cells. I mean, that's what they look like, not a big deal. So what they do is they make these cells and they put them onto a special medium, okay, and this takes a lot of time, a lot of people, and they test they separate the cells into single cell units. They let the cells grow into colonies, and they test the antibodies that colony produces until they find a colony that does what they want it to do. And that takes a lot of work, let me tell you, having spent some time in the lab. All right, and then from that, once they have established which colony is making the right monoclonal antibody that they want, then what they do is they put it into a machine which allows them to replicate these cells in very high concentrations. These things are called bioreactors, and these bioreactors then turn out lots and lots of very specific monoclonal antibodies, and bioreactors are not small or cheap pieces of equipment. They look like that. Okay? So that's a bioreactor that's pumping out the toxin at the rate of 20,000 liters. Okay. So that's the state of the art at the moment, subject to change in next month's issue of the Journal of Immunology. Okay, so what are the advantages? All right, so the advantages are many. Number one, it's a homogeneous antibody. Okay, so as I said, they selected the colonies very specifically. They separate every damn cell into all these little wells, and they check every single colony, see what antibodies it's making, to find the one that best works against their target cells. So it's a very homogeneous thing, which is good and bad. We'll come back to that. It's specific. In other words, the product of that single hybridoma reacts with the same epitope. An epitope is a genetic target. It's a fancy word for genetic target on the antigen. All right, and it need not be pure. Okay, so they don't. What this means is that they don't have to understand the structure of the antibody for it to work. All right, it works very well, and that it's able to they're able to generate, because of all the different colonies that they make, they're able to generate a wide range of antibodies against a lot of different antigenic determinants. And they pick the best one, as I said, and then once they get it going, there's virtually unlimited quantities that they can make of this exact same antibody. So the downside of this are also many, and that is that when you're dealing with a monoclonal antibody, the affinity, which means its ability to react to and hold onto a target, is much lower than a polyclonal antibody, which is the way that you and I normally make antibodies. So for example, if we're making an antibody against the flu virus, where our immune system might make 50 different antibodies against different parts of that flu virus. Does that make sense? Whereas if you're using a monoclonal antibody like this, it's only hitting one part of that flu virus. All right, so it's much more specific, but it doesn't have nearly as powerful a specific immunologic effect as if you have a polyclonal attack against this. Just making sense? All right, good. Okay, then because it's a monoclonal antibody, it may not produce the biological in other words, a single monoclonal antibody may not be enough to induce a complement cascade and a MAC formation. All right. Okay, here's another one. Monoclonal antibodies are very structure specific. So if the structure changes, if the three-dimensional structure changes of the target changes just a little bit, the antibody won't be able to find it and attack it. Antibodies can sometimes unexpectedly cross-react with unrelated antigens, which is a problem, to say the least. And then, as you might imagine, the time, money, and effort is very large, which is why they charge ungodly amounts of money for some of these drugs. But these...
How do they patent it if they don't know the structure?
They're able to patent the process.
The process is known.
Okay, they've patented, but not the antigens that they use, okay, and not the cell clone that they use.
So someone could do something very similar.
And they do. And they do. That's why in that list before you'll see 4 different antibodies against CD20. Okay, and you'll see that again. There are different companies that make different antibodies against the same target. It's not exactly the same, but they're able to do that for that very reason. And that's their problem. Okay. All right. Now, these antibodies aren't just used against cancer, okay? I mean, I know you've all heard of Remicade and Humira against arthritis and colitis and Crohn's disease. One that I use from time to time, this one been listed, which is helpful against lupus and so on and so forth. They even have a monoclonal antibody now against some enzyme that has to do with high cholesterol. So, whatever. Okay. So what I'm going to do now is I'm briefly going to talk about different types of monoclonal antibodies because they all work in a very different way, and I want you to get an idea of how these things can work. Because, as Tom pointed out, there are multiple antibodies sometimes against the same kind of target, okay? But the mechanism of action is going to be very similar, and it'd be a waste of your time for me to spend time on each one of those 4. So I divided this up into basically 5 different classes where basic immune reactions, and I have a representative antibody to talk about each one so you get an idea of how this works. Do you need a short break or can I keep going? Keep going? Good. Okay. All right. Yay, team. Okay. So the first one is Avastin. Okay. Bevacizumab. Avastin. Avastin is a monoclonal antibody which came to be discovered based upon research. Again, I take a historical view of these things because then I can understand better when new things come in, how they build upon the old things, and also how some of the older technology can interact with the newer one and actually give better results. So back in 1940, they noticed that there was a factor X that was released by ischemic tissue and under hypoxic conditions, which led to neovascularization. All right, and what they further found was that discussion was that the molecule, there was a molecule from bovine pituitary follicular cells promoted proliferation of endothelial cells. So this molecule went on to be known as vascular endothelial growth factor, VEGF, okay? And in 1992, it was hypothesized that the original molecule that they were looking at, the factor X, was in fact the same thing as the VEGF. Okay, so physiology of neoangiogenesis, new blood vessel formation, and that is that you have a initial there's a stimulus, there's an outpouching of endothelial cells will generate a new branch, so to speak. The branch will form it'll form like a balloon sort of figure first, and then it'll gradually elongate out into a tube. And this why did you do that? Go away, thank you. And the reason that this happens is because there is a paucity of nutrients that the cancer is getting. It's running out of nutrients. Cancers have very high metabolic rates and need lots of nutrients, and as a result of that, they will start to produce VEGF as a way of stimulating new blood vessel growth to feed themselves, okay? And this is one of the vulnerabilities of solid tumors, is that they rely upon an ever-growing blood supply for them to grow and develop into full-blown cancers. So what happens is that there are many steps that go on in terms of the activation, as I just said, invasion into the tissues, and so there are matrix metalloproteinases that are involved, migration cells that proliferate, sprout formation, and ultimately forming into functional capillaries. So what we have here is is just a quick little diagram of that, and hopefully something that's a little easier to digest. So we start off here with the endothelial cell, and then the endothelial cell is stimulated by the angiogenic factor production by the cancer cell, that's VEGF. Ultimately, you have these sprout formations, you have the extracellular, the endothelial cellular membrane remodeling, growing into ultimately new functional blood vessels that are able to pump more nutrients into the growing tumor. And that's vascularization. This is a picture showing some of the little sprouts and the little balloon-like things that are under the microscope of these blood vessels that are starting the process of neovascularization. So some of the things that can stimulate the production of these angiogenic factors, of course, is VEGF. Here's a friend from earlier, okay, TNF-alpha, which brings us back to the discussion this morning about inflammation. Okay, you all with me? So when tissues are inflamed, back in the old days, they used to talk about pH, and that, cancers were an acidic pH, and acidic pH makes cancer grows. I mean, and translated into current medicine, we're talking about inflammatory and inflammation. The more inflamed something is, the more these things happen. Okay, so TNF is obviously a hugely pro-inflammatory molecule. We're going to talk a little bit more about that in a few minutes, but nonetheless, that stimulates a lot of these angiogenic growth factors, including VEGF. And here's another one that we spoke of before, interleukin-8, which is the SOS cytokine that cancers release. So when cancers are starting to outgrow their blood supply, okay, or the cancers are getting stepped on by a chemotherapeutic agent or some radiation or something and are dying, one of the things they do is they release, why did they do that? They release interleukin-8. And interleukin-8 is the SOS molecule which will stimulate the stem cells in the tissues immediately bordering the cancer to produce more interleukin-8. So you have a whole new area, a whole new milieu of cytokines that are stimulating the production of these angiogenic factors and causing release of VEGF and stimulating blood vessel growth into this area, which is obviously counterproductive for our patients. This is a very good article. That's why I put the PubMed ID in red. Okay, there's only 2 red PubMed IDs in this whole presentation. That's one of them. And what this is looking at are various hallmarks of solid tumors, and it goes into great detail of the different things that can stimulate and support each of these activities. The article isn't so much about therapy, it's just about observation of what happens. And from observation comes hypothesis of how to interrupt that process, and then we have theories, and then we have new drugs, and medicine hopefully then moves forward. But nonetheless, the article lays out in a very nice, readable way how this whole process occurs, and I just thought I would share that with you. It's a relatively recent vintage, as opposed to some of my references from the last millennium. Okay. So hypoxia, of course, is a common theme in cancers, and of course, cancers don't like oxygen, but with hypoxia, when tissues become hypoxic, they also tend to wind up with various nutritional deficiencies, and that's what tends to stimulate the cancers more than the oxygen tension in the tissue. And it goes through a whole series of pathways, ultimately resulting in VEGF and angioneogenesis. Now, there are a number of ways to intersect this. Obviously, mTOR is a major mediator of this whole process, and there are mTOR inhibitor drugs and then that are rather toxic, and then we have metformin, which is rather non-toxic and works well to shut down that mTOR pathway as well. Okay, so we have angiogenesis is critical for the progression of a solid tumor, and this point, the second point, is very important. Most tumors in humans persist in situ for a long period of time in an avascular and quiescent status, years. So then it becomes a question of what turns it back on. What turns it back on, according to the research of Witcher that I was presenting this morning, the thing that gets things going in the wrong direction are two things, okay? The two things are, number one, the pits and inflammation, and number two, as we get older, remember that curve of the immunosenescence that I showed you, our immune system leaves us more and more vulnerable and less able to keep these things in situ as our immune system moves on in time. So it's a combination of these factors as to why, as we age, we have an increased risk of developing malignant disease. Of course, the older we get, the more toxins we've been exposed to and accumulate, so that comes back to the pits. Also, as people get older, their food and tastes often change. If you've ever seen what they serve at some of these restaurants for the senior dinner at 4 o'clock in the afternoon, it's sort of like brown, tasteless, and needs an identification card to know what it is. And the same is true of any nursing home you've ever walked into, I'm sure. So nutritional status can be an issue, but the accumulation of inflammation and the decrease of the production of naive lymphocytes by the thymus are major factors for why this occurs. And this is just back to that. I don't want to bore you with that. Okay, so now we have a study that was done, and there have been endless studies that are done with all of these substances. One of the big groups that does these studies is ECOG. It's the Eastern Cooperative Oncology Group, and they are a big oncology organization that does clinical trials. A high percentage of studies that you read in the medical journals are ECOG studies, and this is looking at progression-free survival. And all the ECOG studies are done with independent third-party facilities, which is one of the reasons that people like to use them. And what they showed was that if you take paclitaxel and mix it with a little Avastin, that people live basically twice as long. And for that sort of reason, the FDA approved Avastin for the treatment first of colorectal cancer. So it's a humanized monoclonal antibody. That's a Zumab, okay? And the way that this antibody was well chosen because it's active, and this is important, it's active against all of the isoforms of VEGF, of which there are 4. There are 4 different subtypes of VEGF. And the way that they built this one, was just lucky because they didn't have the technology back then to know, but they actually built an antibody that hit all 4 of the isoforms of VEGF. So it was a stroke of luck, and it's helped a lot of people. And then the flip side of that is that because of the effect of Avastin by robbing the blood supply to the cancer can cause a cancer to shrivel very quickly. And if somebody has a squamous cell lung cancer, one of the problems with squamous cell lung cancers is that they like to invade blood vessels because they go right for the heart of the matter of getting all the nutrients they can get as quick as they can. But they have a whole secondary blood supply too. And the thing is that if you knock out that secondary blood supply to a squamous cell lung cancer, what will happen is the squamous cell lung cancer can shrink very, very quickly. And if it shrinks quickly and it happens to have also erode into a blood vessel, so for that reason, Avastin is not recommended for people with squamous cell lung cancers. Just as a little aside. Now, the flip side, of course, is that they're still doing tons of research on the use of Avastin, even though Avastin's been around for a while. If you look at the date on that, you'll see this is last month, okay, and here they're doing research on brain cancer, showing that, in fact, it is still helpful and still working and still doing good things for lots of people. Let me see if I can do this. Excuse me 1 minute. But nonetheless, so that was obviously a commercial word from our friends at Roche. I thought that at this point of the presentation, it would be nice to hear a couple of little short segments and give your brain a little bit of a rest, get a little perspective on things. What it didn't say was that, number one, that the regrowth of blood vessels when they remove Avastin occurs because the epigenetic factors, the milieu, intracellular milieu around the cancer has not changed. If in fact you change it by reducing inflammation, then you will in fact reduce blood vessel regrowth, and you will reduce blood vessel growth to begin with. That's number one. Number two, as you're doing a therapy that directly kills the cancer, whether it be IPT, whether it be immunotherapy, whether it be radiation, whether it be anything that directly is killing the cancer, that's going to have a dramatic effect of reducing blood vessel stimulation and growth as well. But for what it was, I thought it would be an interesting little diversion for you. More to come. Okay, next monoclonal antibody we're going to talk about is Ch6. I love Ch6. Ch6 is a monoclonal antibody that does a wonderful job of blocking the bone resorption that occurs as a result of a metastatic lesion to the bone. And the way that this works is that there is, the normal activity is that the osteoclasts are designed to break down bone through a normal process of remodeling. Osteoblasts follow along behind it and rebuild the bone. And through a normal process of bone turnover that all of us go through every single day. Now there is a receptor site that's on the osteoclasts, and that receptor site is also on the osteoblasts to a smaller extent, which would shut them down. And what that receptor site is a receptor site called rank. So rank is a receptor, you're going to love this, it stands for receptor activation of NF-kappa B, rank. Okay? So this brings us back to what we were talking about this morning with NF-kappa B. And this is a key process in bone metastatic development and growth to the point of fracture. And basically, there are ways of blocking the activity of that receptor for NF-kappa B. The first thing that we have to address is are all the things we discussed this morning are things that will help shut down NF-kappa B. This brings us back to a discussion of inflammation. You're starting to see how this stuff is all coming back together and it's all fitting together, and I wasn't purposely trying to bore you to death this morning, but I was showing you where this is coming back. So when you see, this ad and you see this literature in your office and you see rank, it's like, what the hell is rank? Okay, who cares? Well, Ch6 works. Well, we do care because we don't want to just use Ch6 because Ch6 works great, but it's not the only thing out there that can stop this process and help the patient heal. So the rank L part stands for ligand. Okay, remember ligand is an immune term for key. Okay, so basically when you have the key into the receptor and you activate the osteoclast and shut down the osteoblast, you have progressive bone breakdown without regeneration because of the growth factors that the cancer is making to do that. Xgeva was approved by the FDA as a result of this study. And, you know, again, I hate these studies because they're single-agent studies, and that's not the way biology works. But nonetheless, it showed that the bottom line was here: 51% of patients had responses lasting at least 8 months, and so on and so forth. But basically, when they show an agent that has a demonstrable effect compared to patients who aren't treated, then they tend to approve that agent for that purpose. Now, this is showing that the agent by itself was able to stop the bone metastasis activity separate and apart from treating the underlying cancer. Okay? So when I treat a patient with breast cancer, or treating a patient with lung cancer, or treating a patient with prostate cancer, and they have mets to bone, yes, I use Xgeva, but you better believe I'm using everything that I can that makes sense to go after the primary cancer. Okay? I'm also doing everything I can to help shift their levels of inflammation and shut it down. I'm tracking all those blood tests that I showed you earlier. So the ESR, the HSCRP, TNF-alpha, interleukin-1, etc. I'm watching all of those things to see which pathways their immune system is using to generate the inflammation that is causing this process to progress. Because sure, we've all had patients who were model patients and who changed their diet, who took their supplements, who tracked their blood tests, their inflammatory markers crashed, then their cancer stopped or went into remission. Most patients aren't that good. Most patients, quote unquote, cheat. Okay, they aren't as consistent with their life and lifestyle. They go to bed at 2 o'clock in the morning. They get stressed out. They come home from work and have 2 martinis and God knows what, and then wonder why their cancer is not getting better. Hey, so be that as it may, even in that situation, the Xgeva still worked. Because when the FDA does these trials, they do not adjust for lifestyle.
Jesse, are you using this with bisphosphonates as well?
No, I'm just using this these days. Because it works faster than the biphosphonates. There is a risk of osteonecrosis, but it's much less. And I could use this for a much, much shorter period of time. Back in the good old days when I can get IV clodronate, then that was a biphosphonate that worked really well, that also had very, very minimum adverse effects. And then when that became no longer available and I started to use some of the other ones, I was less impressed by those results. But as soon as Xgeva became available, I latched onto that and haven't gone back.